So if you're feeling in the mood to do something really disgusting, open one of the incubators and stick your head inside. Ooh, they're bad. Anaerobes are nasty stuff. Well, I will take mercy on you tomorrow morning. I will come in early and pull out the anaerobic jars and take them outside and open them up and take the things out, because the smell of those is really disgusting. So tomorrow what you're going to do is you're just going to record all of your results, and particularly for your unknown, you're going to get some valuable information. Since you're sharing resources in terms of inoculated known organisms, make sure you pass those around so everybody can document those. Okay? Any questions? Yes, ma'am.
Someone just gave me back my lab manual, so is anyone missing one?
Well, actually, I'm missing one.
Oh, then I guess it's yours.
I missed that with bookstore sticker on it, so if it doesn't have a sticker on the back, then that's probably that.
It says 2017.
Yep.
I'm sure how that happens to that part.
Okay, if you're a microorganism, well, if you're any organism, but since this is microbiology class, what are you hoping to get out of your metabolism? What's the role of metabolism? What should it provide you? Energy, typically in the form of ATP. And just need energy to grow and divide and make new cells and stuff. You need carbon skeletons too, right? So you can do biosynthesis and make new cell membranes and new proteins and nucleic acids and things. And ultimately, what is happening during metabolic reactions? Well, really, what metabolism is all about is moving electrons/hydrogens around from one compound to another. We've got compounds that donate hydrogens and electrons, and we've got compounds that accept them. So metabolism is a lot of oxidation reduction reactions. One of the things I'm trying to impress upon you is that microbial metabolism, in addition to satisfying the needs of particular organisms and keeping them alive, has a bigger, more global role to play as well. And that is really evidenced in the various nutrient cycles for key minerals like carbon, nitrogen, phosphorus, sulfur, etc. And these global cycles of moving nutrients from a reservoir to one part of the environment to another part of the environment, ultimately though returning them back to the reservoir so we have a true cycle, is all dependent upon microbial metabolism. And there are multiple organisms involved in these complex nutrient cycles, and if you take any one of those types of organisms out, you can short circuit the whole cycle. And if you short circuit the whole cycle, life on Earth is in real problem. And so this, again, kind of reinforces the importance of microorganisms in terms of the whole fabric of life. So when we talked about the nitrogen cycle, we said there are 4 general types of microbial metabolic reactions that kind of drive this particular process and really constitute that process of the nitrogen cycle. And they're nitrogen fixation, ammonification, nitrification, and denitrification. So what's accomplished during nitrogen fixation? What happens to nitrogen gas? What's it converted into? Ammonium, okay? And ammonium is a key plant nutrient. That's something that can be absorbed by the roots of plants, taken up and used in the biosynthesis of new plant biomass, making new proteins, new nucleic acids, etc. And you can only do nitrogen fixation if you have this special enzyme, nitrogenase, which some bacteria have. Very few have it actually. Plants don't have it at all. And so this relationship that is developed between microorganisms and plants is really important in providing plants with key nutrients. Okay? So we get this ammonium produced as a result of nitrogen fixation. Okay? Ammonification. Well, that produces ammonium as well, but by a different strategy. Ammonification is really a type of decomposition, and specifically what's happening here is that amino acids are being deaminated, and when the amino acids are deaminated, we're left with organic acids, which can then be fed into the Krebs cycle or glycolysis and can be used as a source of energy to drive microbial metabolism. So the ammonification process takes the nitrogen that was originally pulled out of the atmosphere and converted into plant or animal biomass and releases it back into the form of ammonia again, which could then be taken up by other plant roots and support further plant growth. But ammonium can also be the substrate for another set of metabolic reactions, the nitrification reactions. And during the nitrification reactions, ammonium is becoming oxidized into nitrate. So some of the electrons of this particular compound are stripped off and used to make ATP, and nitrate is the end product of those nitrification reactions. So what does denitrification involve? It involves taking nitrate, okay, and during the process of anaerobic respiration, making things like nitrite or ultimately nitrogen gas. Where does this nitrate work in anaerobic respiration? This is going to be the terminal electron acceptor in the electron transport chain. Okay. So electrons are going to be fed off to the electron transport chain, eventually picked up by nitrate, and nitrate will be reduced into nitrite as one possibility. Once you have nitrite, other organisms can use the nitrite as a terminal electron acceptor and electron transport and reduce the nitrite into nitric oxide. Other organisms can then reduce nitric oxide into nitrous oxide, and other organisms can reduce this into nitrogen gas. So there's a whole set of organisms involved in this process of denitrification. And the product of one organism basically is used as a substrate or an energy source for another organism. As I said before, we're going to do this test in the lab because this is going to be an important test to see if your organism has the ability, particularly to do this type of denitrification, converting nitrate into nitrite. So we're going to do a test to determine specifically whether your organism has that particular capability. That's called the nitrate reductase test. So these two particular diagrams, I'm not going to go through them, you can look at them, just kind of show you how these various metabolic processes that we've talked about interact with one another in an ecosystem to make this thing that we call the nitrogen cycle. And again, it's really important to understand that the cycle has some type of a starting point where the reservoir of material is then converted into other substances, but ultimately the cycle has got to regenerate the starting compound again. So nitrogen gas moves through the ecosystem in various forms of nitrogen, but ultimately nitrogen gas is regenerated again. This involves the interaction of several different types of organisms, plants, bacteria, bacteria interacting with other bacteria, and these particular metabolic processes really are what drive this complex biochemical cycle upon which life depends. So it's a good example to show you not only kind of individual types of metabolic strategies. Okay, so these guys, for example, remember, were the lithotrophs. These are ones that use an organic compound as a source of energy and dump electrons to the electron transport system to make ATP. In this particular strategy of denitrification, an inorganic compound is used as a terminal electron acceptor in the electron transport chain. So this isn't the source of the electrons, this is where the electrons are picked up after electron transport. So different types of strategies could all mesh together to form this critically important cycle that all life on Earth depends upon. So metabolism isn't just about individual cellular needs. These metabolic reactions come together into this process on which all life depends. And I think that's important to understand. Okay, so let's take a look very briefly at the other side of the coin. Let's take a look at phototrophs. Up until now, we have been talking about organisms that are obtaining their energy sources from chemical compounds. Most of them getting it from organic compounds. A few of them, the lithotrophs, getting them from inorganic compounds. The source of energy is chemical bond energy. In phototrophs, obviously, the source of energy is light. Now, remember, to characterize organisms, we not only have to define them in terms of where do they get their energy sources, but where do they get their carbon sources as well. And remember, there are two strategies. If you get your carbon from an organic source, then you are a heterotroph. If you get your carbon from an inorganic source, CO2, then you are an autotroph. So when we talk about phototrophs, we're going to have to talk about both photoheterotrophy and photoautotrophy to kind of get a big picture of what's going on. What's also a little bit confusing about phototrophy is there are different strategies that have been employed by bacteria to get energy from sunlight. Okay? There is oxygenic photosynthesis, and there is anoxygenic photosynthesis. So we need to dig into that a little bit and make sure you understand what's going on. It's believed, and there's a lot of evidence to support this, that the first metabolic strategy that evolved was chemotrophy. And that phototrophy was a later evolutionary advancement. A necessary evolutionary advancement, if you think about it, because we believe that life evolved on this planet primarily in water environments, and organisms evolved within what is sometimes called an organic soup, where the water was infused with all of these organic molecules that had arisen by physical processes, actually. The interaction of radiation with gases in the atmosphere, the interaction of heat with gases in the atmosphere. There's this whole process that we believe happened called chemosynthesis, where organic molecules formed on the surface of the planet and eventually accumulated in the oceans and other bodies of water. And that first cellular forms were all chemotrophs, and they fed on this organic soup. But what would eventually happen? Well, the conditions which at one point on the planet produced these organic molecules no longer existed. Okay, that was a unique time in the Earth's history when this chemosynthesis of organic molecules was taking place. So now you got the soup and you got all these cells that have evolved to start using that as a food resource. But eventually, as those cells multiply and multiply and multiply, what's going to happen to your organic soup? It's going to be used up, right? It's going to be exhausted. And so the food resources would have eventually run out. They were finite in terms of their existence. Think what an opportunity sunlight presented to life on Earth. Another source of energy, which, unlike the organic soup, was pretty much inexhaustible. Now, granted, we said we think the sun's going to burn out in about 4 billion years. But an energy source that is billions of years in existence and has the potential to last that long is pretty much inexhaustible as far as life is concerned. So if organisms could evolve the ability to transfer the sunlight energy into chemical energy, then they didn't have to worry about their food resources running out. And so the evolution of phototrophy was really, really important for a number of reasons. But for the first time now, we had organisms that were the producers of energy, or the converters of energy. They were able to convert sunlight energy into chemical energy. All organisms in the past, prior to the evolution of phototrophy, were living on organic molecules produced by physical processes. Okay, so you see the distinction. So evolution of phototrophy led to having producing organisms in our ecosystem. Well, one strategy that is used by some microorganisms that are actually chemotrophs, so they're using chemical energy as a source of energy primarily, but some chemotrophs have evolved the capability to supplement the energy they get from chemical compounds using light. And this is not technically photosynthesis. This is kind of a precursor to photosynthesis. This particular strategy involves the use of a compound called bacterial rhodopsin. And some bacteria have this, others don't. Primarily, there are chemoheterotrophic purple bacteria that tend to be pretty rich in this bacterial rhodopsin. So what is this stuff? Well, this particular substance is a protein that has very extensive alpha helical regions of secondary structure. And the bacterial rhodopsin is inserted into the cell's membrane. And associated with the bacterial rhodopsin is a substance called retinal. This is basically the same compound that absorbs light in your retina of your eye and allows you to have vision. Retinol has a lot of double bonds. And things that have double bonds have a tendency to absorb light energy pretty readily. If you look at chlorophyll, chlorophyll's got a lot of double bonds. Retinal can exist in both cis and trans configurations. And when it absorbs light, it flops over from the cis condition to the trans condition. And when retinal changes conformation, that changes the conformation of the bacterial rhodopsin. So basically, light triggers this protein via this retinal intermediate to change shape from one shape to another. And that shape change of bacterial rhodopsin does something interesting. It is associated with pumping hydrogen ions out of the cytoplasm and into the surrounding media. So light basically activates this proton pump. And what happens when you start to accumulate protons out in the external environment? Well, if you've got an ATPase channel protein in the membrane, if those protons flow back through the channel protein back into the cytoplasm again, you're going to make ATP. So this is basically a light-driven proton pump which allows you to make ATP. Now, the yield isn't really great. And that's one of the reasons why the typical strategy is that the organisms that do this are primarily chemoheterotrophic and use organic molecules as energy sources. But in the presence of light, they can supplement that energy with some sunlight energy as well. Okay. You can see how that might have been an early strategy that evolved and may have been the precursor to the more complex strategies of photosynthesis. Okay, in photosynthesis, we have a very different setup than we just showed you in the bacterial rhodopsin. We have photosystems. And remember that photosystems are clusters of pigment molecules. Chlorophyll is the main one we talked about in cell biology. There's some other photosynthetic pigment molecules we'll talk about in microbial photosynthesis. So here's our typical scheme of oxygenic photosynthesis. And this is the type of photosynthesis that we've already discussed when we discussed the cyanobacteria. Their photosynthetic pigment is a little bit different than chlorophyll, but basically they photosynthesize the same way plants do. Remember, two photosystems, and the purpose of those photosystems is to absorb sunlight and convert it into chemical energy. The two major forms of chemical energy we need to be produced are ATP. Well, that's produced by this hydrogen ion gradient that's produced during electron transport. You may or may not remember that the electron transport system connects photosystem 2 and photosystem 1. Well, we make ATP using light energy. But we need something else to reduce CO2 into carbohydrate. We need some hydrogen. The source of the hydrogen ultimately comes from water. The hydrogens are stripped off of the water, basically, and ultimately transferred to a coenzyme, NADP. And NADP is produced during this photosystem number 1 over here. So we get ATP, reduced NADP. And if we then have CO2 available, those three things can interact in the Calvin cycle to basically make carbohydrate. Okay? So we're converting light energy into chemical energy using some light-absorbing pigments clustered together into these photosystems. So that's oxygenic photosynthesis, because when the water is split, oxygen's a byproduct. So we talked about the significance of oxygenic photosynthesis in terms of changing the face of the planet. Oxygen's needed to make ozone. Ozone layer filters out UV light. UV light's damaging the cells. The evolution of photosynthesis allowed the transition of organisms from aquatic environments to terrestrial environments. We also need oxygen to support aerobic cellular respiration, which is much more efficient than fermentation, and was a crucial feature in the evolution of multicellular organisms. So oxygenic photosynthesis was a big thing. Other strategies that were used by bacteria were a different type of photosynthesis called anoxygenic photosynthesis. Anoxygenic photosynthesis differs in a couple of important ways from oxygenic. And one of the ways that they differ is in the type of pigments that are used. In oxygenic photosynthesis, we use primarily chlorophylls. And this top figure right here shows you the absorption spectrum of chlorophyll. And what you see is that you get a nice healthy absorption at the blue and violet wavelengths. And then you get this real dip in the green, because chlorophyll is reflecting those wavelengths of light. And then we get another spike in absorption at the red wavelengths. In anoxygenic, there is a different compound that's used as a photosynthetic pigment. called bacteria chlorophyll. And this is kind of a reddish purple type of pigment. Now, if you look at the absorption spectrum of bacteria chlorophyll over here, we still get some absorption in that blue wavelength region, but not as heavy as we got with regular chlorophyll. We still get that dip in the green regions down here. Look at the red. The red doesn't absorb particularly well, especially compared to chlorophyll. But then we get out here into the longer wavelengths of light, the near infrared and the infrared. And again, we get a big spike in absorption of light at those particular wavelengths. Important thing, do you remember the relationship between wavelength and energy as we move to higher wavelengths on this spectrum right here? Does your energy increase or decrease? So does this have more energy over here, or does this have more energy over here? Higher wavelengths, longer wavelengths like the IR, near IR, have less energy than the shorter wavelengths. That's actually going to be significant in a minute. I'll come back and talk about that in a minute. Okay, so we talked about anoxygenic photosynthesis when we did our little survey, and we talked about the green sulfur bacteria and the purple sulfur bacteria. One of the ways in which anoxygenic photosynthesis differs from oxygenic is that this strategy only employs a single photosystem. As it turns out, this particular photosystem is photosystem number 1. So this is oxygenic, 2 photosystems connected by an electron transport chain. Anoxygenic, a single photosystem. Light is absorbed by the bacterial chlorophyll. Electrons in bacterial chlorophyll are excited to a higher energy level. And those electrons are picked up by compounds and eventually donated to NADP, producing that reductant we need for the Calvin cycle. Well, this isn't sustainable, because ultimately what's going to happen to those electrons in bacterial chlorophyll as they get boosted up and taken away? We're going to deplete the electrons in that pigment. It's going to become oxidized. So we need a place to replace those electrons from. In oxygenic photosynthesis, where do those electrons come from? They come from the splitting of water. In anoxygenic photosynthesis, they come from something else. Very typically, reduced sulfur compounds like hydrogen sulfide. Sometimes from hydrogen gas. Sometimes from some organic molecules like succinate. Succinate is one of the intermediates of the Krebs cycle. But these molecules are split. Okay, here's a little bit of application now. Why would it be difficult for this absorbed near IR light to split water effectively? It doesn't have enough energy, okay? These longer wavelengths of light that are being absorbed are lower energy wavelengths of light, and consequently don't have sufficient amount of energy to break those sulfur-hydrogen bonds, those SH bonds. I didn't know what those values were, but I just looked at them before I came. The bond energy of an SH bond is 81, I believe, and an OH bond is 111. So there's significant more energy needed to break the OH bonds, and that is supplied by absorbing higher energy wavelengths of light in oxygenic photosynthesis. Images.
Sorry, what was the question you asked?
What was the question you asked?
Why is it difficult? Why there was more absorption?
Oh, why isn't it possible for this wavelength of light to be involved in oxygenic photosynthesis? So can the energy absorbed by bacterial chlorophyll split water, basically? The answer is no. The energy that's absorbed is not high enough. Now, as this water is split, as this hydrogen sulfide is split, or hydrogen is split, or succinate is split, a proton gradient is formed, which allows us to make ATP. So we've got our two essential things we need from the light energy. It's being converted into ATP and reduced to ADP. But it only took one photosystem to do it. Okay, so these are photoautotrophs now. In both cases, they're using CO2 as their carbon source. In both cases, they're using sunlight as their energy source. But one strategy produces oxygen as a byproduct and uses two photosystems. The other strategy doesn't produce oxygen and uses one photosystem. But we've still got, I shouldn't erase it, under phototrophs, we've still got these organisms that use organic carbon. So these are photoheterotrophs. And this is kind of a bizarre strategy if you think about it. Photoheterotrophy is another form of anoxygenic photosynthesis. So it's only going to use a single photosystem again. It's carried out by different types of bacteria. The oxygenic photosynthesis we talked about in photoautotrophy was carried out by purple and green sulfur bacteria. The photoheterotrophs are purple and green non-sulfur bacteria. There's something else interesting happening here. There is not an external electron donor involved in this process. So there's not water. There's not H2S. There are not molecules being split to generate electrons. This particular bacteria chlorophyll is a little bit different than the bacteria chlorophyll here. This one absorbed near IR light. That's a little bit shorter wavelength than infrared. This one absorbs infrared radiation. This absorption, though, has enough energy to boost an electron up to a higher energy level. But it's unstable up there. So what's it going to tend to do? It's going to tend to fall back down to its lower energy level again. But when it does so, the electron passes through an electron transport chain. And so it generates ATP. But what doesn't happen is NADP is not reduced. Because in the previous strategy, that electron was grabbed by NADP and reduced it. So we supply new electrons to the photosystem from an external compound. Here, the electrons are just going bup, bup, bup. This is called cyclic electron flow. And it does generate ATP. This one, whoops, is called non-cyclic. Okay, the electron doesn't go back here. It's grabbed by something else. This is non-cyclic electron flow as well. So is that a problem? Well, yeah, you'd think it would be, because don't you need a source of reductant of hydrogen to reduce CO2 into carbohydrate? Yeah, if you're an autotroph. But these are heterotrophs. Okay? So they're getting their carbon from organic molecules. They're not producing it by Calvin cycles. So this is a little bit different strategy. One of the things I would like to kind of impress upon you is that microorganisms have been around for billions of years. And evolutionarily, just about any strategy to survive that anybody can even conceive of, and probably some we can't even conceive of right now, has been tried in the microbial world. Most weren't successful, but some were successful and have hung on, and organisms have persisted that have used strategies that we look at now and go, that's bizarre. This is a bizarre way to survive. Okay? To be an organism that uses sunlight to make your energy, and an organic molecule to make your carbon. Okay? And all these different strategies have evolved, and that's what makes the microbial world so interesting and complex, particularly from the standpoint of metabolism. Now, there's one other interesting thing that I need to throw on this to increase your level of complexity even a little bit more, and I'll try to keep it as simple as I can. There are a set of organisms, and one particular interesting organism I'm going to highlight, that are said to be facultative photoheterotrophs. And that word facultative should start ringing some bells for you. Okay, facultative anaerobes, what can they do? They can do aerobic respiration when oxygen is present. They can do anaerobic respiration when oxygen isn't present. So what does a facultative photoheterotroph? It depends upon the environmental conditions. In anaerobic environments, these organisms can do one thing. In aerobic environments, they might do a different thing. Or in the presence of light, they might do one thing. And in the presence or the absence of light, they might do another thing. One of the things that breaking organisms down into metabolic categories suggests is that a particular organism only uses one strategy. So if you are a photoautotroph, that's all you ever are. Or if you are a chemoheterotroph, that's all you ever are. But in reality, there are organisms that are adaptable and can employ one strategy under one set of conditions and switch over to another strategy in another set of conditions. So light present, oh, I can be a photoheterotroph. Light not present, oh, I can be a chemoheterotroph. So that is what we're talking about when we talk about facultative phototrophy or facultative chemotrophy. There's a really interesting organism called R. palustris. This purple guy right here. Okay, this is a purple bacterium. And this is a facultative photoheterotroph. Does one thing in the presence of sunlight, does another thing in the absence of sunlight. It's incredibly sophisticated metabolically. Again, the driver seems to be presence of light, absence of light. Okay? These are pictures of these little purple bacteria. Don't look very impressive. But they can do some really cool things. This bacteria is sometimes referred to as the bacterium that eats anything. This particular organism can utilize a variety of different substrates for both carbon sources and energy sources. And many of these compounds, as it turns out, are important environmental toxins. And so these particular organisms have a lot of interest by people working in bioremediation, using microorganisms to degrade environmental pollutants. So these organisms can live on these toxic substances and thrive using them as energy sources and carbon sources. And they can do very interesting things like fix nitrogen. Well, that's a real bonus. Okay, nitrogen fixation is a really important process. So man, something that could degrade environmental pollutants in the soil and add nitrogen to it at the same time? Wow. That's pretty cool. And many of their metabolic reactions generate hydrogen gas. Biofuel. That's pretty cool. Researchers in this particular area envision a time when these type of bacteria could be seeded into wastewater treatment plants and could degrade sewage and produce biofuel as a product. Think about how many sewage treatment plants we have in the country. And how if we could turn those into hydrogen generating biofuel plants, what a neat cooperative system that would be. The beauty of this particular organism is its capability to carry out a multitude of different types of biochemical reactions, and the fact that they are this facultative strategy where one set of conditions they do one thing, the other set of conditions they do another thing. And that really helps to illustrate to you some of the sophistication that occurs within certain types of microbial metabolism. Okay, so that's the ending point as far as this section is concerned. We've got another exam coming up a week from Monday, so we've got some more material to cover. The next thing that we're going to talk about, which is already posted, has to do with the fact that we have been talking quite some time now about how microorganisms grow, and metabolism was an important part of that growth process. Now we need to turn the corner and talk about how do we kill microorganisms? Why might we want to kill microorganisms? Because for all the good they do, there are a number of microorganisms that are pathogenic. When we talked about the history of microbiology, we talked about how shocked people were when von Leeuwenhoek discovered animalcules present in what looked like to be clear, pristine water samples. And as that realization came to people, especially in big cities like London in the 1600s, people started to think, man, if these samples of water that look fairly pristine are teeming with microorganisms, what does something like the smelly Thames River contain? Because the Thames River was still a major source of water for the citizens of London, and it was also the primary sewage treatment plant. Untreated sewage ran through the streets and ran into the Thames River. And so this shows you a caricature of someone looking through a microscope at Thames River water and being aghast because people suspected that these particular organisms might be causes of disease. And so once the organisms were discovered, immediately people started to say, how can we control them? And that was particularly relevant because people knew that diseases were contagious, and a contagious disease like the Black Plague that existed 300 years earlier had wiped out like half the population. So even before people knew microorganisms were the cause of disease, they knew that there was something that was causing disease to spread. And some interesting strategies developed, particularly in the developing medical field, to try to prevent doctors and other healthcare people from getting sick as they treated patients. And so garbs, for example, like this one, were developed to try to shield people from contamination by infectious disease. And much of the strategy just involved covering, but I think this little beak-like mask is kind of interesting. This contained materials like cotton to filter the air that you were breathing in, because the idea was that there was something in the air that was causing illness. Other strategies were used, like burning various aromatic woods and incenses. One of the things that people understood even back in the Roman era was that generally speaking, people who drank from metal cups, copper, silver, gold, had a much lower chance of getting waterborne illness than people who drank out of wooden cups. All this anecdotal evidence started developing about strategies to control the spread of microorganisms and to kill pathogens. And many times the method became fairly well established long before anyone understood what the mechanism behind that method was. So there is a method, there's a mechanism behind how copper and silver and gold retard microbial growth. There is a kind of a logic to why burning certain types of incenses, for example, could retard microbial growth. Some of the woods contain formaldehydes, for example, and those were released when the incense was burned. So this information about how to control microbial growth has kind of accumulated over a long period of time, and slowly we're kind of starting to understand not only what methods work, but what the mechanism behind some of those methods. Now, what we're going to do is we're going to break down microbial control methods into 3 categories. We're going to talk about methods of physical control, chemical control, and in methods that are considered to be chemotherapy. My goal is to get here by end of week. So this is the next section in terms of notes posted, PowerPoints posted, would be the general physical methods and general chemical methods of control. There is some terminology that you need to understand. So when we talk about control methods, I'm going to throw around words fairly commonly that involve using things like the terminology pasteurization and sanitization and degerming and aseptic and antiseptic, sterilization, static, stasis, cycle. And so what I'd like you to do prior to Monday is to look through this particular slide and make sure that you understand what these are. I'll elaborate just a little bit on a couple of them, but ultimately you've got to understand the lingo to be able to converse with this particular material. There's another slide that I really urge you to look at and maybe even print out or keep with your notes or whatever. But this is kind of a flow diagram that breaks things down logically into various categories that we'll talk about. Okay, so we'll talk about physical agents, and we're actually going to lump together physical agents and mechanical agents together. And then we're going to talk about chemical agents as well. So this is going to be a good kind of organizing tool to talk about some of the stuff, we're going to talk about next week. Okay, so tomorrow come in. When you take your thioglycolate tubes out of the incubator, move them back to your desk carefully. You don't want to jostle those tubes because you don't want to disrupt the growth that's in there, because you want to see the growth patterns. So that's what we'll be doing tomorrow.